Bismillahirrahmanirrahim. So now we will start with the chapter number 3, that is epithelium. Epi means upon and thelium means nipples. So epithelium is kind of word derived from these two root words. Now, what are tissues of the body? As we know, cell is the basic structural and functional unit of life. When cells combine together, they form a tissue. These cells are related to each other because they are similar cells specialized to carry out similar function. There can be different types of tissues. They can be epithelial ones, they can be connective ones, muscle ones, nervous ones, and tissues of the body. Different tissues, they combine together to form organs, and each organ has two type of things. One is parenchyma and other is stoma. Parenchyma, since it is starting from P, so it is performing specific functions, whereas stroma, which is starting from S, so it is the supporting tissue of an organ. So now we move towards epithelium, which literally means upon the nipple. So it is a collection of closely opposed cells which are present very close to each other, and they have a very limited amount of the extracellular matrix. The epithelium is avascular. It means it does not have any large amount of blood vessels, and it is connected by loose connective tissue. They have a basement membrane or basal lamina to provide support to them, and this basal lamina is composed of glycoproteins and collagen type 4 substances. Now, the main substances which are required for the functioning of epithelium, they're supplied to them via diffusion from the blood capillaries beneath the basement membrane, because the epithelium actually lacks any blood vessels. So now the oxygen, carbon dioxide, and metabolic waste, they are exchanged with the blood capillaries of the basement membrane via the process of diffusion. Now we have two types of epithelium, which is a glandular epithelium and the other is the covering epithelium. Now, covering epithelium will actually cover the external surfaces of the body, whereas the glandular epithelium, it will actually make the glands of the body. Now, the glandular epithelium, it can do two type of function. One is secretion and other is excretion. So secretion is a word that is used for the removal of the useful substances or release of the useful substances, and this release is done by the process of exocytosis, and the secretions are released in the form of secretory granules. Now, the excretion, that is the removal of the waste products like metabolic waste, like carbon dioxide, like urine, these are removed via the process of residual bodies. They actually are present in the form of residual bodies which are moving out of the cell by the process of exocytosis as well. Now we have the covering epithelium. It is actually a lining epithelium. It will cover the external structures of our body, and it can be multilaminar or it can be unilaminar. Now we will study the types of epithelium in detail. So now the epithelium is further divided into three types of epithelium. One can be simple, which is unilaminar, meaning so simple epithelium is unilaminar, which means it has only a single layer of cells, whereas stratified, it is multilaminar. It means it has many layer of cells, and pseudostratified is pseudo means false and stratified is many layered. So it is actually a single layered type of cell, but it gives a single layer type of epithelium, but it gives a idea or a false accusation that it is stratified. So now the simple epithelium, it is actually further divided into a simple squamous epithelium, simple cuboidal epithelium, and simple columnar epithelium. Squamous means it is composed of flat cells. Cuboidal means it is composed of cube-shaped cells which have same length and same width, and columnar means it has a tall length but a short width, these type of cells. So we can see it over here. We have the simple squamous cells, beneath which we have the basement membrane or the basal lamina, the simple cuboidal cells which are cube-shaped cells, and beneath which we have the basement membrane, basal lamina, and beneath which we have connective tissue. And then we have the simple columnar cells which are tall column-shaped cells, and they have a basement membrane, and beneath the basement membrane we have the connective tissue which has a large number of blood capillaries within it. So now we'll look at the features of simple squamous epithelium. It shows that their cells are flattened, they are polygonal in shape, they have serrated margins, a flattened nucleus. All of it I will show you in the diagram in a moment. Flattened nucleus located in the center of the cell, and beneath them we have the basement membrane. So as you can see in this diagram, we have flat polygonal cells with a flattened nucleus centrally located, and they have serrated or wavy margins, and these margins fit into each other, and this is how the simple squamous epithelium is present, beneath which we have basement membrane, beneath which we have a connective tissue layer. And now the thickness of this simple squamous epithelium is 0.1 micrometer or less. The simple squamous epithelium is present in endothelium, that is the internal lining of the blood vessels, in the type 1 alveolar cells which are located in the lungs, and the mesothelium, that is in the pericardial, pleural and the peritoneal cavities. The main function or the main substances, that exchange of the substances that occurs in the simple squamous epithelium is by the process of diffusion, but in some location active transport and vesicular transport can occur, but mainly it has diffusion. Now look at the simple cuboidal epithelium, which are square in shape. They have the same height and width. Their cells are hexagonal or polygonal, and they fit into each other. They have a spherical nucleus which is located in the center of the cell, and example are epithelium of the thyroid gland and the follicles, epithelial lining of the follicles of the thyroid gland and the distal convoluted tubule of the kidney. Also, they can be present in the ovary. So they can be in the thyroid, in the kidneys, and in the ovary. So you can easily visualize the simple cuboidal epithelium having cube-shaped cells, beneath which is a basement membrane, beneath which is a layer of the connective tissue, and these cuboidal shaped cells are present in thyroid gland follicles, in the ovary, and in the kidneys. Now the simple columnar epithelium, which are tall cells having shorter width, and they have an oval shaped nucleus which is located near the base of the cells. Example of simple columnar ones are in the gallbladder, intestines, stomach and uterus, and they serve to increase the surface area. So simple columnar cells, they are only a single layer as they are simple, and columnar, they are tall having shorter width, a circular nucleus which is located on the basal side, and these cells are located in gallbladder, in the uterus, in the stomach and in the intestines. And in some location of the cells they might have cilia, kinocilia, stereocilia and villi on their surfaces. Also look at these two diagrams. These are actually how they appear in the microscope. So you can easily see that the cuboidal shaped cells, they can be visualized over here, which are having same length and same width. So these are simple cuboidal ones which are located in thyroid gland and in the kidney tubules. These are the tall columnar cells, as you can see. These are simple columnar epithelium which can be found in the stomach, intestine, gallbladder and uterus. And you can see the cells are very tall, having a basal nucleus, and beneath which we have the connective tissue. So now we come towards the stratified cells, the stratified epithelium. The stratified epithelium has many layers of the epithelium. On the basal side, it has tall columnar or cuboidal cells, which are actually mitotic ones, and the cells, they gradually become polygonal and they become flattened to the surface. So columnar or cuboidal, which are mitotic, then they become polygonal, then they become flattened or squamous on the external surface. The stratified squamous can be keratinized or can be non-keratinized. In the keratinized one, the external surface has keratin filaments, and the cells, their desmosomes, they gradually die. Their desmosomes are not present and they flake off from the surface in keratinized cells that is present in the epidermis of the skin. But in the stratified squamous non-keratinized epithelium like this one, the cells on the surface, they do not flake off. There is no keratin layer and they are present in oral cavity, esophagus and the oropharynx. So now we visualize this stratified squamous keratinized epithelium. So you can easily see a large area of the keratinized area, and the cells, they are flaking off and they're losing their desmosomes. Whereas we have this stratified squamous non-keratinized, and in this area we see the cuboidal shaped cells which become polygonal and then flattened on the exterior surface. They do not lose their desmosomes, do not flake off, and they are present in oral cavity, esophagus, and the oropharynx. The basal region has mitotic activity, and the mitotic cells, they move to the top layer, in which they undergo from the cuboidal to the polygonal to the squamous transition. And since they undergo this transition, they are also known as the transitional epithelium. So here we have the stratified cuboidal epithelium, which has 2 or 3 layers of the cube-shaped cells which are stacked over each other, and beneath them there is the basement membrane, and beneath them we have the area of connective tissue with the blood capillaries in them. Now, these stratified cuboidal epithelium are present in the ducts of some glands like pancreas, salivary glands, and the duct of sweat glands, and the function is absorptive function or secretory function. Always remember that cube-shaped cells have absorptive or secretory activity, and they are present in the glands. This will also help you in studying embryology very well. So now we have the stratified columnar epithelium. It consists of a large number of column-shaped cells which are stacked over each other, but the basal layer is of cuboidal cells which are having a mitotic activity. And this stratified columnar cells, they are present in conjunctiva of the eye and the male urethra, but they are not replaced by the basal mitosis. This is one feature, that they are not replaced by the basal mitosis, and there is no progression from the base to the surface. So now we have the transitional epithelium or urothelium, which lines the lower urinary tract. This urothelium, it is present in renal pelvis, calyx, ureters, urinary bladder, and the main function of this epithelium is to undergo stretching, that is distension and undistension, stretching and relaxation. So now the main features of undistended or relaxed one is it is having 6 or more layers, and basal is of cuboidal cell as always. Above the basal we have the polygonal cells, and above the polygonal cells we have the umbrella shaped cells which are convex and may contain 2 nuclei. Now the distended one, it is actually the contracted state, and it has 2 or 3 layers. The basal layer can be cuboidal, above it the polygonal cells, and above it the superficial most layer consists of the umbrella shaped cells. Let me show you. So you can clearly see the transitional epithelium in undistended bladder. It has 6 or more layers, basal layer being of cuboidal cells, above it the polygonal cells, above it the umbrella cells. The umbrella cells might have 2 nuclei, like in this one. Now we have this transitional epithelium in distended urinary bladder. It is contracted one. You can see the lower cuboidal, they have lost their cube shape, but still we can appreciate them as cuboidal. Above them the polygonal, and above them we have the umbrella shaped cell that might have 2 nuclei. Since these cells can undergo a change in the number of their layers, so they are mostly involved in the functioning of contraction and relaxation, like in the urinary bladder, which has to contract to excrete the urine out of your body, or it has to be distended to store the urine inside the body. And same goes to the renal calyx or renal pelvis, in which a large amount of water might be stored or water might be removed from the body of the kidney. The transitional epithelium has a large number of other features as well, like it is impermeable to salts and water. Secondly, these cells are strongly bound by zonula occludens. We studied that zonula occludens does not allow any paracellular loot, so it does not allow any leakage of the material, and these cells can stretch and contract. One other feature of the transitional epithelium is that they have plaque region and interplaque region in the umbrella shaped cells. That is, plaque region are rigid cells and they cover an area of 12 nanometer, and then they are interrupted by flexible cells in the interplaque region which are 8 nanometer in thickness. These ones are 12 nanometer in thickness and they are rigid and they are plaque region, whereas these are 8 nanometer in thickness and they are interplaque region and they are flexible. This is how the whole transitional epithelium looks like under the microscope. Let me show you again. So transitional epithelium having basal cube-shaped cell, above it the polygonal cell, and above it the umbrella cells, which can be binucleated or mononucleated. Now we have the pseudostratified epithelium. This can be ciliated or non-ciliated. So pseudostratified epithelium has short basal cells and tall columnar cells. And these short basal cells have their nucleus in the lower area, whereas these tall columnar cells have their nuclei in the apical region. So they give an appearance of a false stratification, false multilayer, but actually they are single layered. These tall columnar cells, they have a basal region attached to the basement membrane and the luminal region which is attached to the is present in the luminal side. Whereas these short basal cells, they only have basal surface, but their apical surface does not open into the lumen. They have cilia on their apical surface or luminal surface. They are mainly present in the nasal cavity, nasopharynx, and the trachea and bronchi. Main function of these cells is mucociliary transportation. Simple as that. They might also be present in male genital ducts like the ducts of epididymis, but they have stereocilia or non-motile cilia in the male genital ducts, whereas they have kinocilia or motile cilia in the respiratory passages. In the respiratory passages, they have to perform the function of mucociliary transportation. In the male genital duct, they do not have any function because they are non-motile, or they can be in the signal transduction or transfunction. Now we move towards epithelial, two special variety of epithelial cells. They are neuroepithelial and myoepithelial cells. I have drawn their shapes as well. So the neuroepithelial cells, they are present in tongue, in the taste buds, or in the ear, that is the vestibulocochlear system. Main function of the neuroepithelial cells is to conduct the nerve impulses to the central nervous system. So now the anatomy of these neuroepithelial cells or histology is that they have an apical surface with cilia, microvilli, and stereocilia, and on the basal surface they have an afferent nerve fiber that is attached to them, and they carry the sensory impulses to the central nervous system. Now these myoepithelial cells, these are star-shaped cells, and they are present in the and they contain a large amount of actin and myosin filaments because they have to do the function of contraction, right? And these myoepithelial cells, they are located in the ducts of glands, and the main function of these is to allow the contraction of the glands, to allow their secretion to be expelled out of their main body. This is the function of myoepithelial cells. So in the previous chapter, in the previous video, we have a video where I have told you about the zonula occludens, adherents, and desmosome. That was the junctional complex. Now the gap junctions and this whole area, it actually makes the lateral domain of a cell. Above it, we have an apical domain on the apical side, which has stereocilia, the non-motile one, kinocilia, the motile one, and microvilli. And on the basal side, we have a basement membrane, and beneath the basement membrane, we have the basal lamina. And also, there are infoldings of the cell at this area of the basement membrane. Now, and the cell has this nucleus and all the other organelles on the inside. Lateral side has junctions, apical side has cilia or microvilli, and the basal side has basement membrane and connective tissue, etc. So firstly, we will study microvilli, which have mainly absorptive and oscillatory function. These microvilli, let me show you. These were located above, and now these are here. These microvilli have a plasma membrane outer covering, and on the inside they have a large number of actin filaments. These actin filaments are linked to each other by the fimbriae or fascin molecules, and beneath them we have the actin filament attached to the terminal web, which has myosin and spectrin molecules and intermediate filaments. The terminal web has myosin, spectrin, and intermediate filaments, whereas this region has actin filaments linked to each other by fascin and fibrin. These are present in villi, that is the main area of small intestine, and they serve to increase the surface area, and they are present in the proximal convoluted tubule of the kidneys. Looking at them, we see them as small cylinder or finger-like projections. They are covered by plasma membrane. They have 20 to 30 microfilaments or actin filaments. Actin filaments linked to each other by fascin and fibrin. The actin filaments are attached down to the terminal web. In the terminal web, they are attached to myosin and spectrin, and beneath the myosin and spectrin, we had the intermediate filaments. They make striated border in the intestines, and they make brush border in the proximal convoluted tubule of the kidneys. Function: increase the surface area, and they have absorptive function. Also, oscillatory contractile movement of the microvilli facilitates in the absorptive function. So now my most favorite, that is a internal structure of cilia. So now cilia is 1 to 2 micrometers in length. 5 to 10 micrometers in length. 1 to 2 is the microvilli. And cilia is 5 to 10 micrometers in length, and in diameter it is 0.2 micrometers. It is covered on the outside by a plasma membrane, and this is the structure of an exoneme of the cilia. It has 2 singlets in the center and 9 peripheral doublets. These peripheral doublets, these have dynein arms, which are actually having ATPase activity. They are having a radial spoke and a central 2 single microtubules, which are covered by this plasma lemma sheath. So now the functional signals, they come through this area, travel via the radial spoke to this peripheral region. The dynein arms, they do their ATPase activity and get the energy from ATP, and they crawl over each other and they are attached to each other. This linkage is the Nexin one. As simple as that. And these dynein arms, they help in crawling of the doublets over each other, as a result of which the cilia can contract or relax and do the effective stroke and the relaxation stroke. They can do both of it. So now we have these 13 protofilaments. They are A and they are B. 13 protofilaments are present in the A1, but the B has 10 protofilaments and it shares 3 of them with the A. So it has 13 in its cell as well, but 3 of them are shared with the A1, but actually B has a total of 10 protofilaments. They're also saying that they are made up of microtubules, and these microtubules come from a basal body, and basal body has 9 plus 0 pattern. It does not have two central molecules or 2 central tubular passages. And the peripheral 9 doublets, they become continuous with the 9 triplets microtubules of basal body. So as I told you, when the kinocilia, it stiffens the dynein arm, they crawl over each other and they do the effective stroke, which allows the movement and bending, and recovery stroke, in which it moves to its original location. Main function of the cilia was in mucociliary transportation or mucociliary clearance. It is a protective function which is done in the nasal cavity. Whereas the kinocilia, this is the function of the kinocilia or the motile cilia. They are also present in the neuroepithelial cells of the internal ear and the tongue, and they allow in the transduction and the mechanical transduction of the signals or the mechanical stimuli into electrical impulses. Like they can convert the mechanical signals into electrical impulses which are carried to the brain or the central nervous system. So now we have a disease which is primary ciliary dyskinesia. It is also known as the Carter-Jenner syndrome. It is an autosomal, it does not depend on the sex chromosome, but is an autosomal recessive genetic disorder, and it affects the motility of flagella in the males, the flagella of the sperms in the males, as a result of which the patient cannot have the mucociliary transportation, so they have upper and lower respiratory tract infection. Also, they have infertility in males because their sperms, their tails of the sperms cannot move, and hence these type of things occur in primary ciliary dyskinesia. So now we have the primary cilium. It is also known as the immotile cilia. It is involved in signal transduction only. The stereocilia, which are the non-motile cilia, they are thin, they are hair-like, and they are numerous in number. And at the apical portion, they actually intertwine with each other like this, and they form a tuft-like appearance. As a result of which, they are incapable to move, and they are mainly located in epididymis and vas deferens, that is the male genital ducts, and might also be present in the neuroepithelial cells of internal ear. Main function is main function is to increase the surface area to facilitate in the reabsorption process or in the conduction of the mechanical impulses into electrical impulses and transmission of these impulses to the central nervous system by the neuroepithelial cells of the internal ear. Lateral domain has all the type of cell junctions which allow cell to cell and cell to basal lamina communication. All of that I have taught in the previous video. You can see that video. Now we have the basal domain, and in the basal domain we have basement membrane, the hemidesmosome which link the cells to the basement membrane, and the infoldings of the plasma lemma. Let me show you. So we have infoldings of the basement membrane itself, and the hemidesmosomes. So we have a very important diagram to discuss over here. This is the diagram of the basal area. The basal area of the cell has hemidesmosome, a large number of infoldings, and then lamina lucida, lamina densa, and the reticular lamina. So the lamina lucida is an electron lucent layer, and it is 40 nanometers thick. It has laminin and intactin. Moving towards lamina densa, it is 50 to 60 nanometers thick. It is a little bit dense layer. It has collagen type 4, laminin, and perlecan. Beneath this is the sublamina or the reticular lamina. It has glucose aminoglycans, which are actually chondroitin sulfate, keratin sulfate, heparin sulfate, and these are collagen fibers actually, and these are collagen type 7 fibers. Collagen type 4 over here, collagen type 7 over here, no collagen over here. And it has GAGs, that are glucose aminoglycans. Main function of the basement membrane is to attach the epithelial cell with the connective tissue. Connective tissue is a layer that is present beneath the basement membrane. So the basal lamina, it allows the basement membrane to be attached to the connective tissue layer. Then it has a large number of scaffolding which are very effective in wound healing process. Then they allow growth, proliferation, and differentiation, and they also act as ultra filter that allow the passageway of some molecules, but they impede the passageway of other molecules. For the function of epithelium, I have a mnemonic that is PASTS. P is for protection, A is for adhesion, S is for secretion, T is for transportation, and S is for sensory reception. This is absorption. So protection, absorption, secretion, transportation, and sensory reception. And the protection, the keratinized, squamous keratinized epithelium, the epidermis, which protects the body against drying and soaking and bacterial invasion. Secretion, like the glandular secretions or the secretory surfaces. Adabsorption, Some epithelial cells like the microvilli and the cells that are located in the stomach, etc., they allow absorption. Excretion is of the waste substances, urine and sweat, etc. Transportation is also done by them by the mucociliary transportation, as you know, in the ciliated epitheliums. And neuroepithelial cells, which are located in the taste buds and in the internal ear, they are involved in sensory reception. Also the stereocilia, they are also involved in the sensory reception. This was all from the chapter number 3, that is the epithelium. I hope you understood each and everything. If you have any query, any question, please let me know in the comments. And if you understood anything, please like, comment, and subscribe to my channel. Thank you very much.